Okay, so yeah, we have been talking in this Congress about cancer sometimes and some other times about contagious diseases. So I am here to join those two fields in this talk. So yeah, I will be talking about contagious cancers, also known as transmissible cancers. But before we deep into that, let's remember some key basic concepts of cancer and evolution. So, in a very simplified way, cancer starts with one or a bunch of cells mutating and they start to divide in an uncontrolled way, forming a primary tumor. Eventually, one of the cells of that tumor that is heterogeneous will acquire the ability to abandon that primary tumor and disseminate to other organs or tissues of the body. This is what we call metastasis. Cancer nowadays is the second death of course worldwide, well, if we only take into account high-income countries, and it is popular knowledge that the sooner you get a diagnosis of cancer, the better prognosis you will have, the more chances of survival. But why does this happen? And it's exactly because of what we were talking about, the metastasis. The metastasis is estimated to be responsible for 90% of those cancer deaths. And this is because most of the treatments focus on eliminating the primary tumor with little emphasis on metastasis. Therefore, if we study with innovative ways metastasis, we will be able to illuminate universal processes, and there will be new opportunities to develop new cancer therapies. So that's why we are very interested in understanding metastasis. Another thing is that usually when the patient dies, the cancer dies with it. In other words, the cancer is restricted to the life of the host. But this is not the case for all the cancers, because sometimes the cancer cells can spread to other individuals. This is what we call clonally transmissible cancers or contagious metastases. And as you can see here, they can live beyond the death of the host. And not only that, if you think about it, this process is not that different of the metastasis. It's just a metastasis at a large scale, and that is contagious. So, these transmissible cancers or contagious metastases are particularly very frequent in the marine environment, more specifically in bivalves such as cockles, which are the ones that we are going to study. So cockles have a leukemia-like cancer. And if we take a look at the hemolymph, which is like the blood that we have, but for these animals, which you can see in the syringe, you can see 3 cell types that are healthy cells. But sometimes in some individuals, we see those big, round cancer cells. And those are the cells that we like to study. To do that, as common cockles are distributed from Senegal to Russia, we collected and diagnosed cockles from 11 different countries, and 5% of those cancers were diagnosed with this leukemia-like. So we had the samples, but we were interested in studying the genetics of these cancers. So another milestone to do this project was to assemble and annotate the reference genome for cockles. So we did that, and we ended up with a chromosome level assembly of the common cockle. And just a fact, the genome size of cockles is 0.8 gigabases, which is like a third of the human genome, but within the range of bivalve genomes. So we were prepared. We need to sequence cancer cells and healthy cells to be able to ask questions and investigate the contagious metastases. So to do that, we sequenced almost 600 samples composed by tumors, also by healthy cells from the individuals where we took the tumors, and healthy cockles to understand the genetic structure of the cockles. So we were in a good position to start asking questions like how many times did contagious cancers appear in cockles? Where did they appear? Where are they found? Or where are all of them contagious? So to answer this last question, let's see how could we do it with phylogenetic approaches. So in the case of a non-contagious cancer, if we take cells of a single individual, tumor cells and healthy cells, like we did here, and we do this with several individuals, we will find that the cancer cells will cluster next to the healthy cells of that individual, because the cancer was originated in that individual. But if we are facing a contagious cancer, all the cancer cells in different individuals will have been originated in a single cockle. They will have a common ancestor. So instead of those cancer cells clustering next to the healthy cells of that individual, all the cancer cells will cluster together, as you can see in this picture. So we did that, and this is the result. So as you can see, the tumor samples, the hemolymph of those cockles, cluster with hemolymph of other cancer cockles, while the healthy cells of those cockles are clustering in the middle of the phylogeny. So we are facing contagious cancers. We have no example of non-contagious cancers in our sample collection. Another thing that you can see is that cancer in cockles appears to have been originated at least 9 times. As you can see, 9 clusters of cancers. So we had already answered 2 of the questions that we were questions that we were making at the beginning. But we were also interested on where have these cancers been found and where have they arisen. So to answer the second one, we focused on the sister taxa. This is the samples that cluster nearby the cancer lineage in the phylogeny. So what we can see is that some cancer lineages extend thousands of miles, like you can find them in Spain and Ireland, for instance, while some others are found just in one place, like this one in France or this one in Ireland. So the distribution is very different, and it depends on the cancer lineage. And where have these cancers arisen? Well, this question is very difficult to answer, but we do have some clues, because as you can see, some of the cancer lineages cluster mainly with samples from the north, while some others, like for instance this cancer in France, cluster with healthy samples from the south. So there is some difference among the cancer lineages. So finally, you are all scientists, so you all know that you do research and you to answer a couple of questions, and you open 10 more questions. So these cancers in cockles have been studied for a long period by histological methods, and they were classified into types, type A and type B. And when we had a look in our samples, which were type A and which were type B, as you can see, there is no mixture of type A and type B in any cancer lineage, whether they belong to type A or to type B. So this opened the questions of why do we find these phenotypes, histological phenotypes, and is it regarding the nuclear DNA? Because here we have been studying the mitochondrial DNA. So our next step is to study the nuclear DNA to see if they tell us the same story. So last but not least, I would like to thank the organization committee of this conference, because they give me the opportunity to talk to you today about contagious cancers, and also all the scientists, collaborators, funders and institutions that allow us to do this research. So thank you very much.